Okay, we'll start today. We'll see the remaining part from your general pharmacology. In the last session, we have seen this part. We have seen about the general pharmacognosy last session. We have seen about the traditional system of medicine. So that part is important. They may ask you one question from that part. So after that, we have also seen some active important constants from plant origin which are used in the therapeutics. We have also seen this secondary metabolites of plant. I will explain that part again. We have seen the classification of core drugs. We have seen this part also. We have seen these chemical groups, etc. etc. Plant tissues. We have seen types of stomata. In the last session, we have finished our types of stomata, trichomes. In the last session, we have seen this drug adulteration process. We have also seen microscopic evaluation of crude drugs. So what do you mean by stomatal index? What do you mean by stomatal number? What do you mean by palisade ratio? So that is again important part. So just remember stomatal index of a plant. Stomatal index of a plant, that property will remain constant throughout the life of, because that stomatal index is nothing but the, it is a ratio, and that's why it will remain constant at all. So stomatal index is nothing but the S divided by E plus S. What is S? Number of stomata. And what is E? So this E, S is nothing but the number of stomata, and E is nothing but the number of epidermal cells. So this stomatal index property of plant that will remains constant throughout the life of plant. You have to remember this. Okay. After that, we have seen different leaf constant. Palisade ratio means number of average average number of palisade cells beneath the each epidermal cell. Epidermal cell के नीचे leaf constant. These are known as a leaf constant. Okay. So जो drugs which are, or crude drugs which are containing leaves, or where the crude drugs where there is a biological source involves leaf. So in that case, palisade ratio is important, when islets number is important, when termination number is important, stomatal numbers is important, stomatal index is important. These are the qualitative properties of a particular plant. So for example, a authentic plant of digitalis will have stomatal number in a range. Authentic plant of your digitalis or belladonna will have a particular palisade ratio. Authentic plant will have a definite vein number, okay, definite vein termination number, definite vein islet number, so that you can identify the adulterant by using this. So these are nothing but the microscopic evaluation parameters for a crude drugs. There are other also, but these are important for your exam. Stomatal index is equal to S divided by E plus S into 100. What is S? Number of stomata per square mm. Number of stomata, okay, then E plus is number of epidermal cell per square mm plus stomata into 100. This property of plant remain constant throughout the whole life of plant, because it's a ratio. We have seen this. We have seen the lycopodium spore method. So just remember lycopodium spore method. It is a inexpensive method with official test status, and it is used to measure the percentage purity of drug. The lycopodium spore method is a quantitative microscopic quantitative method for purity of drug. This was question in GPAT 2023. So what is the purpose of this lycopodium spore method? So it is a quantitative method for measurement of percentage of purity of drug. ड्रग कितना प्योर है, परसेंटेज प्योरिटी ऑफ ड्रग माइक्रोस्कोपिकली. तो लाइकोपोडियम स्पोर मेथड, ठीक है, जीपैट 2023 में क्वेश्चन आ चुका है. So again, each lycopodium spores, they are very uniform in nature, and each spore has a size of 25 micron. इसके ऊपर क्वेश्चन एक बार आ चुका है. 1 mg of lycopodium spore consists of 94,000 spores. 1 mg lycopodium पाउडर में 94,000 spores होंगे. ये कांस्टेंट होता है, एंड दे आर द एक्सट्रीमली यूनिफॉर्म स्पोर्स. एक्सट्रीमली फ्लेमेबल पाउडर होता है. इट इज नथिंग बट द वेजिटेबल सल्फर obtained from club mass of lycopodium clavatum family lycopodaceae. We have already seen this. but I have revised because इसके ऊपर बहुत बार क्वेश्चन आ चुका है. So remember, it is a method for measurement of percentage purity of drug. Lycopodium spore. Okay. So in case of ginger also, in case of ginger, this is a constant 286000. You can measure or you can quantify the if any foreign matter is there in a powder drug. आपके पास में बेलेडोना है, बेलेडोना का पाउडर है. बेलेडोना के पाउडर में स्टार्च ग्रेन्स आपको मेजर करना है. If you want to measure a starch grain, particular characteristic structures, you can use lycopodium spore method. एक ऑयल रहता है, ऑयल लाइकोपोडियम एंड वो जो पाउडर ड्रग है उसका एक पर्टिकुलर अमाउंट में ऑयल में डिसॉल्व करके you have to observe under microscope. You have to count particular characteristic structures that might be of starch grains, that might be of foreign particles, that might be of your particular characteristic oxalate crystals. So you can decide percentage purity of drug by using this microscopic method known as a lycopodium spore method. Okay, this method is important. इसके ऊपर 2-4 बार क्वेश्चन आ चुका है. जीपैट में आता है, नाइपर में आता है, 100% क्वेश्चन आता है इसका. Am I audible you clearly? Okay. Next is nothing but the WHO parameter for standardization of herbal drugs. WHO parameter. Okay. WHO parameters for standardization of herbal drugs. So you should know. Look, यार मूर्खान से. So you should know the authentic drug. So there are a lot of parameters which can be checked. So there are, for example, organoleptic characters like color, odor, taste, texture. You can check that. You can check its microscopic characters like types of calcium oxalate, type of stomata in case of leaves, vein islet number, ventilation number, parasite ratio in case of leaves. So if you want to find out a particular characteristics structures, you can use lycopodium spore method. There are certain parameters which are fixed and which are observed. For example, volatile oil content of some drugs. जो drugs है जिसमें volatile oil होता है, volatile oil content of few drugs. So it is a constant. For example, in case of caraway, it is a 2.5% weight by weight, not less than 2.5%. So it is an important characteristic for caraway. 2.5%, not less than 2.5% in case of lemon peel also. Lemon peel. In case of clove, 15%. क्वेश्चन इसके ऊपर आ चुका है. What is the volatile oil content? What is volatile oil content present in the clove? Clove मतलब का लवंग होता है. Clove में कितना होता है volatile oil? So 15% weight by weight. In case of fennel, fennel means soap. So 1.5%, not less than 1.5%. Not more, not less than 2.5% in case of dill, and cardamom, not less than 4%. So some drugs also show some fluorescence under UV light. अगर आपने ड्रग्स का पाउडर या फिर क्रूड ड्रग्स का जो भी क्रूड ड्रग का पाउडर और सैंपल, if you analyze that sample under UV, so they are going to show you certain kind of fluorescence. For example, cinchona will show you purple blue colored fluorescence. Gentian root will show you whitish blue colored fluorescence. Ipomema will show deep purple violet color fluorescence. Quassia bark will show you whitish blue color fluorescence. Rhubarb will show you violet colored fluorescence. Wild cherry माओ कलर फ्लोरेसेंस देता है. Arachi's oil मतलब अपना शेंगदाणा तेल. Arachi's oil means groundnut oil, and then sesame oil, they will show blue fluorescence. Epic acona will show you orange colored fluorescence. Aconite will show you light blue colored fluorescence. Gentian light blue. So these parameters they have already observed. Okay? You can check the purity of drug, whether that is authentic drug or not. ठीक है, आपने मार्केट में से क्लो लेके आए हो तो आपने चेक करना है उसमें कि volatile oil कितना है. अगर वो 15% से कम है, so if it is less than 15%, that is not an authentic drug, or that is not a pure drug. Okay? So if you purchase the cloves from market, it should contain not less than 15% weight by weight volatile oil, known as a eugenol. Eugenol होता है उसमें. These are the parameters. There are certain parameters. We'll see WHO parameters for standardization of herbal drugs. Okay. Standardization parameter for evaluation of quality of crude drugs. Today we will see this part, and also today we will try to finish the biogenetic pathway. Biosynthetic pathway in case of plants. Plant में glycosides कैसे बनते हैं, primary metabolites क्या होते हैं, secondary metabolites क्या होते हैं, tracer technique क्या होता है. Okay, so on that part you can expect one or two questions. So after all, they are going to ask you 10 questions from pharmacognosy. You might have seen in your GPAT voucher. आपको 10 क्वेश्चन आने वाले हैं इसमें से, और उसमें के मैक्सिमम क्वेश्चंस तो एक तो एक या दो क्वेश्चन जनरल फार्माकोग्नोसी में से होते हैं. एक आधा क्वेश्चन आपको अल्कलाइड्स, एक आधा क्वेश्चन अल्कलाइड्स ग्लाइकोसाइड उनके ड्रग के बारे में होता है, या फिर उनके टेस्ट के बारे में दूसरा क्वेश्चन आ सकता है, या फिर उनके क्लासेस के रिगार्डिंग क्वेश्चन होता है. So I am going to explain it. So standardization parameter for evaluation of quality of crude drug. So you can evaluate the quality of crude drug by using organoleptic properties. Organoleptic properties मतलब क्या है? Color, odor, taste and fracture. जैसे authentic drug जो होता है जिंजर, उसका कलर कैसा होना चाहिए? It should like a pale grey color. टेक्सचर कैसा होना है, फ्रैक्चर कैसा होना चाहिए. इफ यू सी, अगर आपके पास में कोई सिंकोना बार्क है या फिर अर्जुना बार्क है, तो उसको फ्रैक्चर तोड़ के देखो आप. तो फ्रैक्चर इज आइदर स्मूथ और इट इज अ रेगुलर और इरेगुलर और क्रेनेट. so you can check the purity of that fracture, taste, odor. These are the organoleptic properties. You can check the pharmacognostic properties. Okay, you can check the pharmacognostic properties of your food drugs. Okay, so macroscopic and microscopic. So these properties they are further divided into macroscopic. Macroscopic मतलब ये आपको विदाउट माइक्रोस्कोप आप इसको इवेल्युएट कर सकते हैं, लाइक शेप, साइज, एक्सटर्नल अपीयरेंस ऑफ दैट प्रोडक्ट्स. Microscopic characters they are qualitative, microscopic characters quantitative, microscopic characters and scanning electron microscopy. Okay, correctly. Qualitative, quantitative क्या है? So quantitative microscopy. So in that whatever leaf constants are there. So all these are the quantitative microscopic characters of the plant. Palisade ratio, vein islet number, right? Stomatal number, etc. etc. Okay. Then in case of leaves, leaves में leaf कैसा है? Dorsiventral और isobilateral. Leaf अगर है उसकी मार्जिन कैसी है, लैमिना कैसा है, मिड्रिप कैसा है, मार्जिन में भी बहुत सारे टाइप आते हैं. सो डिपेंडिंग ऑन दैट, जैसे स्टेम अगर ले लिया आपने तो स्टेम कैसा है ना. सो ऑल दीज आर द pharmacognostic parameters. Now you can also check the physical parameters, like for example physical parameters in that moisture content, presence of moisture or presence of water in the crude drugs. That is important because presence of moisture will, okay, presence of moisture will facilitate decomposition of your that crude drugs. अगर crude drugs में water content है, water content क्या है? जब water present होगा तो hydrolysis, जो active content का hydrolysis भी हो सकता है. So for example in case of digitalis, presence of moisture more than 2% will increase the activity of Dg per days, which will cause hydrolysis of glycosides present in the digitalis. So moisture content is important. You can measure the moisture content. Now for the moisture content measurement, you have different methods. For moisture content measurement, you have different method. First one is known as a okay, toluene distillation method. Toluene distillation method. Okay. Second method is known as KF reagent, Kull-Fischer reagent. So Kull-Fischer reagent is the toluene distillation method is there. Toluene distillation method. Okay. Toluene distillation. Kull-Fischer reagent is there, and third is nothing but the IR spectroscopy. So you can measure the water content of crude drug by using these three. Toluene distillation method is there. Okay, then Kull-Fischer reagent is there, and your IR spectroscopy is there. So you can measure the water content present in the crude drug by three. Then extractive value. Okay, suppose extractive value में दो तीन टाइप का आता है. So in that water soluble extractive is there. Okay, water insoluble extractive is there. Okay, then particular type of solvent, organic solvent, let's say ether soluble extractive, chloroforms, okay, ether soluble extractive, alcohol soluble extractive, okay. So it is again a particular characteristic of plant, ठीक है. जैसे मान लीजिए कि आपने जैसे होता है धतूरा का एक्सट्रैक्शन किया, so extractive content of dhatura, it may be either 0.5 to 1%, okay. So most of the cases it is, okay, it is that, say for example 1%. So if you brought another drug, if you do same thing, extractive value in the alcohol, so if it is more than that, that means there is some adulteration or there is some that plant is not authentic. So extractive values, okay. Then ash value, what do you mean by ash value? Ash क्या होता है, ये कोई ash मतलब ये ऐश्वर्या राय वाला ash नहीं है. What is mean by ash value? Ash value means residue which is remained after incineration of particular plant. आपने जो भी है आपका सैंपल, आपका जो सैंपल है उसको क्या करना है, आपको इग्नाइट करना है. यू हैव टू इग्नाइट दैट सैंपल, ओके. सो रेसिड्यू रिमेन्ड आफ्टर द इंसिनरेशन. इंसिनरेशन मतलब क्या है, पूरा उसको जला देना है. इन द क्रूसिबल यू हैव टू प्लेस 1 gram of drug and you have to ignite that, okay. So whatever is remaining after complete ignition of that, that is known as a ash value. So there are different type of ash value. इसमें भी बहुत सारा type होता है. Water soluble ash value, water insoluble ash value, acid soluble ash value, acid insoluble ash value, alkali soluble ash value. इसके ऊपर भी एक बार क्वेश्चन आता है. So for example, acid insoluble ash, insoluble. What is the significance of acid insoluble ash? Okay. So first you have to do, you have to take a sample, 1 gram sample, जो भी ड्रग का आपको एसिड इंसोल्युबल ऐश करना है उसका. तो यू हैव टू टेक 1 gram सैंपल, यू हैव टू इग्नाइट इन द क्रूसिबल. क्रूसिबल होता है उसमें डालकर उसको इग्नाइट करना है. आफ्टर द पर्टिकुलर टाइम यू हैव टू take that crucible, you have to weigh the ash as it is ash, and then you have to dissolve that ash in a acid, okay, acid. You have to dissolve that ash in a either sulfuric acid or hydrochloric acid, and whatever remained, okay, whatever remained, okay, you have to filter, and after filtration whatever you are getting, that is acid insoluble part of that ash. So what is the significance of that acid insoluble ash? उसके ऊपर जीपैट में क्वेश्चन आ चुका है. देखो एसिड में क्या डिसॉल्व होता है, ऑल ऑर्गेनिक मैटर जो है वो एसिड में डिसॉल्व होगा. एम आई गेटिंग, आर यू गेटिंग माय वॉइस? एसिड में ऑल ऑर्गेनिक कंपोनेंट जो है, जैसे प्लांट मटेरियल है, सेलुलोज वगैरह, वो सब डिसॉल्व होगा. बट अगर कोई उसमें इनऑर्गेनिक कंपोनेंट है, से फॉर एग्जांपल सैंड है, उसमें अगर कोई मेटल है, इनऑर्गेनिक कंपोनेंट, sand, okay, inorganic component, so that will not be dissolved in the acid. So if a particular crude drug has a more acid insoluble ash, that means what, it indicates the presence of inorganic content. क्या करता है वो, अगर आपने, सो पर्टिकुलर ड्रग है, जैसे मान लीजिए कि आपके पास में क्या है, आपके पास में सेना है, सेना का एसिड इंसोल्युबल एच कितना दिया है उन्होंने, नॉर्मल वैल्यू दिया है उसका लेस देन 0.1%. So if normal value of acid insoluble ash for the senna, if it is more less than 0.1%, so if you do same thing for drug, whatever you brought from the market, and if you brought senna from market, and if you do the acid insoluble ash, if it is more than 0.5%, what it indicate, that indicate the this sample of senna has a more inorganic content. दिस वाज क्वेश्चन इन जीपैट, ठीक है. तो ये जीपैट के ऊपर इसमें क्वेश्चन है. देन फ्लोरोसेंस एनालिसिस. फ्लोरोसेंस एनालिसिस वी ऑलरेडी इन द लास्ट स्लाइड आई हैव एक्सप्लेन्ड यू. और एक का फ्लोरोसेंस देता है अंडर यूवी. सो ऑर्गेनोलेप्टिक, cognostic, pharmacognostic and physical properties, you can check this, okay. Standardization parameter. Again chemical properties, okay, chemical properties. So there might be either qualitative and quantitative. Qualitative means it is a presence है या नहीं है ना. Qualitative means for example whether a carbohydrate is present in the plant or not, whether alkaloid is present or not, whether glycoside is present or not, है. So that is a qualitative test. So it is done by using HPLC, is known as a high performance thin layer chromatography. और उसमें it is a high performance thin layer chromatography modification of TLC. Okay, modification of TLC in which the mobile phase, then sample application. So it is an automated process. So it is a actually HPLC is a fully automated. HPTLC is a semi-automated. Okay? Semi-automated techniques. HPTLC, high performance thin layer chromatography. So in that there is a atomic development of plate in a mobile phase over development automatic होता है उसमें, and there is one chamber known as a there is one chamber. The detection of that spot or band is done in the one chamber known as a 10 centimeter. 10 centimeter, right? 10 centimeter. So in the HPLC ये विजुअलाइजर होता है 10 centimeter. क्वेश्चन इसके ऊपर आ चुका है. Then you can check the presence of secondary metabolite, like okay, you can check the presence of secondary metabolite, DNA, okay, secondary metabolite, then DNA fingerprinting, then chromatography. Okay, that is a qualitative test for analysis. Now quantitative test means how much quantity is present. Again it is done by HPTLC, GLC means gas liquid chromatography, HPLC high performance liquid chromatography, EFC means fluorescence chromatography. Now this is the chemical test. Now what are the biological and toxicological evaluation parameters? Biological and toxicological evaluation parameters for crude drugs. Biological and toxicological parameters for evaluation of crude drugs. So first, in that biological microbiological भी आता है. Microbiological में total aerobic count, आपका जो crude sample है ना, कौन सी फार्माकोग्नोसी ड्रग में वो क्रूड सैंपल जो है, क्रूड ड्रग जो है, उसमें टोटल एरोबिक काउंट कितना है बैक्टीरिया का, दैट इज आल्सो यू हैव टू क्वांटिफाई. देन ई कोलाई, प्रेजेंस ऑफ ई कोलाई, प्रेजेंस ऑफ साल्मोनेला, presence of enterococci bacteria, presence of alpha toxin. So actually this alpha toxin should not be present in the any crude drug. Alpha toxin crude drug में आना ही नहीं चाहिए. So important है. Then okay, so microbiology में आता है आपका total aerobic count, E. coli, salmonella, presence of and absence of alpha toxin. Then in the pharmacological evaluation, so you can check the bitterness value of that compound. So for the bitterness value standard is the standard, इसके लिए जो होता है, इसके लिए quinine hydrochloride is used as a standard. Quinine hydrochloride. So using the bitterness value के लिए standard होता है quinine. Quinine सिंकोनाल क्लोराइड quinine वाला. Then hemolytic index, for that it is done on a ox blood hemolytic index. So saponins glycosides, they are the standard for hemolytic index. उसके बाद में आता है फॉर्मिंग इंडेक्स. ये सब गुल इज यूज्ड स्टैंडर्ड फॉर फॉर्मिंग, सॉरी फॉर फॉर्मिंग इंडेक्स आल्सो अगेन saponin glycosides are used. For swelling index isabgol ASCII is used as a standard. तो इसके स्टैंडर्ड्स भी याद रखना है आपको, उसके ऊपर भी क्वेश्चन आता है. फॉर द बिटरनेस वैल्यू स्टैंडर्ड यूज्ड इज, so that is a quinine, ये आप मैं लिखता हूँ quinine. For hemolytic index saponin. For foaming index saponin. For swelling index isabgol. Okay. Then toxicological parameter or evaluation. So in that pesticide residue is there and heavy metals are there. So heavy metals के ऊपर क्वेश्चन आ चुका है. We'll see the quality control of herbal drug physicochemical properties. So first one is nothing but the ash value. So ash are of three types. ash जो होता है वो तीन टाइप का होता है. ash मतलब क्या है? What is meant by ash? Residue remained after incineration of the plant material or organic matter. So if you have to burn the organic matter, whatever is remaining, that is known as the ash. So there are the total ash value is there, water soluble ash is there, and acid insoluble ash. So higher the ash value, more with the inorganic content. जो organic content होगा वो तो burn हो जाएगा. So if inorganic contents are there, okay, so that means there is a more ash value. So if particular powder drug का ash value आपको मालूम है कि इसका ash value इतने रेंज में आता है. So if you are getting a more ash value, that means the sample is contaminated. Okay. So acid insoluble ash का भी यहाँ पे याद रखना है. So what is the inference of acid insoluble inorganic content और sands? देखो आप एसिड की बोतल कहाँ पे रखते हैं? सैंड में रखते हैं. सैंड जो बालू होता है बालू. And water content it is can be done by either water content can be measured by either loss on drying LOD, in azeotropic distillation or tolling distillation method, or by KF reagent. In volatile oil measurement, it is done by steam distillation. It is done by steam distillation, and for that particular apparatus is used Clavinger's apparatus. GPAT 2018 में इसका proportion. Which apparatus is used for the isolation of volatile oil from the volatile oil by using steam distillation? Which apparatus is used for the isolation of volatile oil by using steam distillation? Clavinger's apparatus. इसको याद रखना है. ठीक है. Then marker compound. इसके ऊपर एक बार क्वेश्चन आया GPAT 2021 में. What is marker compound? So marker compound, it is used to study the metabolic processes in the plant. ठीक है. प्लांट में ग्लाइकोसाइड बन रहे तो है या नहीं. So marker compound क्या होता है? Marker compound is nothing but it is a compound of known purity. It is a compound of known purity with or without therapeutic activeness. GPAT 2021 में क्वेश्चन आ चुका है. So what is marker compound? Marker compound is nothing but the compound of known purity. Okay? है ना. For example, if you want to quantify, कि आपको क्वांटिफाई करना है कि डिजिटलिस में कितना परसेंट डिगोक्सिन है. आपने जो सैंपल है आपके पास में उसके एक्सट्रैक्ट में डिगोक्सिन कितना परसेंट है. तो उसके लिए आप कैसे कर सकते हो, है ना. उसके लिए आपके पास में स्टैंडर्ड तो चाहिए ना कोई. तो स्टैंडर्ड डिगोक्सिन जो होता है दैट इज अ मार्कर कंपाउंड. फार्माकोलॉजिकल इवैल्यूएशंस bitterness value. So unit equivalent to the bitterness of standardization of quinine hydrochloride. ये standard होती है quinine hydrochloride. So quinine hydrochloride is used as a standard in case of bitterness value. In case of hemolytic activity, हीमो मतलब क्या है? हीमो मतलब ब्लड होता है. लाइटिंग मतलब क्या है? ब्रेकडाउन. ब्लड को ब्रेकडाउन कौन करता है? सैपोनिन ग्लाइकोसाइड. So it is carried out on ox blood and reference solution is nothing but the saponin solution. In astringency, so the property of particular chemical to precipitate the proteins known as the astringency. So fraction of tannins that binds to standard Hyde powder. Astringency is measured by using Hyde powder. Question इसके ऊपर भी आ चुका है. Foaming index. It is nothing but the foam height which is produced by 1 gram of material under investigation. Okay, you have to just take a material जैसे सैपोनिन होता है ग्लिसराइजा या फिर कोई भी सैपोनिन ग्लाइकोसाइड. अगर वो ले लिया आपने, उसका पाउडर ले लिया, उसमें कुछ 5 ml वाटर डालो टेस्ट ट्यूब में और उसको थोड़ा शेक दो, आपको एक पर्टिकुलर फोम आएगा. सो फोम हाइट यू हैव टू मेजर. सो फोमिंग इंडेक्स इज नथिंग बट द फोम हाइट प्रोड्यूस्ड बाय 1 gram of material under specific condition known as the foaming index. Okay? Mostly applicable for the saponin glycosides. Saponin वाले जो होते हैं उसको. Swelling index. So it is swelling index is applicable for isabgol. इसके ऊपर एक क्वेश्चन आता है, what is the swelling index of isabgol? Swelling index कितना होता है इसका? इसका होता है 8. Okay? Swelling index of isabgol 8 to 12 centimeter. इसके ऊपर क्वेश्चन आ चुका है. Swelling index of isabgol 8 to 12. Okay? Toxicological evaluation, in that you are going to have pesticide rescue, because while cultivating the medicinal plant, you are using different pesticides to control the pest on the plant, right? So there should be a limit of pesticide on the crude drugs. Total organic chloride and total organic phosphorus, these are the measures. Heavy metals के बारे में क्वेश्चन आ चुका है. Heavy metals, the analysis of heavy metals present in the sample that is done by atomic absorption spectroscopy. Atomic absorption spectroscopy इसके ऊपर GPAT 2023 में 2 क्वेश्चन आ चुके हैं. So heavy metals, it is tested by atomic absorption spectroscopy, and in case of crude drugs, in case of pharmacognosy, lead, mercury and cadmium. If you know lead, mercury and cadmium, these 3 heavy metals limits are there, and these metals should not be present beyond the limits. Okay? GPAT 2020 में 21 में एक क्वेश्चन आया था कि in case of WHO parameter of standardization, crude drugs, the analysis of heavy metal is done for following. उसका ऑप्शन में एक ऑप्शन था लीड मर्क्यूरी एंड कैडमियम. So heavy metals, okay, the crude drugs are studied for these heavy metals. So they, whether they are present or not, their limit. Okay? So heavy metals, analysis of these heavy metals is done by atomic absorption spectroscopy, and in the crude drugs, heavy metals are detected these three. That is the lead, mercury, and cadmium. WHO guidelines for general limits. So crude drugs should be totally free from alpha-toxin. Alpha-toxin crude drugs में नहीं होना चाहिए. Radioactive contaminant as per the recommended international atomic energy. Okay, recommendation of international atomic energy agency. कि आपके इसमें रेडियोएक्टिव कंपोनेंट है क्या क्रूड ड्रग में, उसका लिमिट एज पर द रिकमेंडेड, ओके, एज पर द रिकमेंडेशन ऑफ इंटरनेशनल एटॉमिक एनर्जी. माइक्रोबियल लिमिट कितना होता है, इसके ऊपर भी एक बार क्वेश्चन आया है. माइक्रोबियल लिमिट इन द प्लांट. सो साल्मोनेला इन द क्रूड ड्रग्स फॉर प्रोसेसिंग, क्रूड ड्रग्स फॉर प्रोसेसिंग, so it should be absence, so it should be absent. And crude drugs for internal use, it should be nil, मतलब absent. Crude drugs for topical use, again nil. Salmonella should be absent. Okay. In case of enterobactery भी नहीं होना चाहिए आपके. Crude drugs for processing, for drug for internal use, उसका लिमिट दिया है 10⁻3. Okay. ये तो मतलब ये मेजर नहीं करती. Crude drug for processing उसमें salmonella मेजर नहीं करेंगे. Crude drug for processing अगर है, so if crude drug is for processing, कोई raw material है, so they are not going to test salmonella है या नहीं देखेंगे नहीं. Okay, but if the crude drug is for internal use, तो उसमें ये salmonella absent होना चाहिए, because ये क्या करेगा salmonella, अगर ये अंदर गया पेट में तो ये डायरिया करेगा, डायरिया will produce diarrhea. So it should be absent for the topical use as well as internal use. Okay, enterobactery should, okay, it is not checked in the crude drugs for processing, but if crude drug is for internal use, it should be within a limit 10⁻3 colonies. For topical use 10⁻4 is the limit. Total aerobic count, अगर crude drug for processing है so it is not done, but if crude drugs for internal use, so total aerobic count should be less than 10⁻5. Total aerobic count should be less than 10⁻7 in case of topical application. If for E. coli, so for crude drug for processing, limit is 10⁻1. Okay. Again for internal use 10⁻1 is limit. Topical use 10⁻2 is the limit. This is the limit for microbial load. Now there are different parameters in the cultivation collection of plant. Cultivation collection जब करते हो medicinal plant का, so you have to exercise certain things कि आपको particular altitude में ही रखना है. So it is given. So altitude required for the plant, okay, type of soil required for the plant, pH of soil, type of irrigation required for plant, कि उसको किस तरीके की मिट्टी चाहिए ना, वो कितने हाइट तक आता है, कितने अल्टीट्यूड पे आता है. ठीक है. उसके बाद में व्हाट इज द टेम्परेचर और एनवायरनमेंट रिक्वायर्ड फॉर द ऑप्टिमम ग्रोथ ऑफ दैट प्लांट. प्लांट की ग्रोथ को टेम्परेचर कितना होना चाहिए सहन करता है, कितना नहीं. सो ऑल दिस कंडीशंस दे आर गिवन इन द कल्टीवेशन collection parameters for the crude drugs, or you can say good agricultural practices. In that also they have mentioned. So it is, everything is not important for us, but you should know. So the, for example, tea plant will grow, जो tea plant होता है Thea sinensis, it will grow in an altitude of 1000 to 1,500 meters. Optimum growth altitude. Cinchona will have 1000 to 2000. Camphor 1,500 to 2,000 meters from the sea line. ठीक है, जो होता है sea line, height from sea line. कहाँ पे cinnamon 250 to 1000 मीटर, and that's why cinnamon देखो इसका altitude क्या है, कम है, और दैट्स वाई सिनेमन जो होता है कहाँ पे उगता है ये, इट इज मोस्टली फाउंड इन द कोस्टल एरियाज। कोस्टल एरिया जो होता है ना वहाँ पे ही ये सिनेमन वगैरह का, जैसे अपने यहाँ पे महाराष्ट्र में कोकन रीजन है, कोकन कोकन रीजन में ये सिनेमन का प्रोडक्शन होता है। सो फॉर द कॉफी अल्टीट्यूड रिक्वायर्ड इज 1000 टू अगेन 2000, फॉर द क्लोव अगेन अप टू 900। देखो क्लोव भी क्या है, क्लोव भी सी लेवल से थोड़ा नजदीक ही ग्रो करता है। और सैफरन 1.25, सैफरन कहाँ पे आएगा, सैफरन आता है कश्मीर में, देखो अल्टीट्यूड कितना ज़्यादा है। फॉर कार्डेमम आल्सो 600 to 1600, it is also grown in the coastal region. Optimum temperature for cultivation है ना, so optimum temperature will also affect the quality of plant, okay, and the quality of active content in the plant. So optimum temperature should be maintained. For in case of cinchona 60 to 75 Fahrenheit temperature is the coffee के production के लिए, if you want to cultivate the coffee plant 55 to 70 Fahrenheit temperature is there. For tea 70 to 90, for cardamom 50 to 100. So they are not going to ask you the altitude, they are not going to ask you the temperature, okay? But you should know this, that's why I have included. Okay? Type of soil, okay, soil type of soil will also affect the क्वालिटी ऑफ एक्टिव कंटेंट फ्रॉम द क्रोटल। आपका जो प्लांट है अगर आपने प्रॉपर सॉइल में रखा ही नहीं, उसको प्रॉपर फर्टिलाइजर ही मिला नहीं, तो आपका जो ड्रग जो एक्टिव है उसमें उसका उसका क्या उसका जो एक्टिव है उसमें क्या रहेगा, उसका क्वालिटी क्या रहेगा। सो ओके, टाइप ऑफ सॉइल इज आल्सो इम्पोर्टेंट। सो टाइप ऑफ सॉइल ऑन द बेसिस ऑफ पार्टिकल साइज, तो फाइन क्ले जो होता है सो दे विल हैव पार्टिकल साइज लेस देन 0.002 mm। ठीक है, कोर्स क्ले और स्लिट जो होता है इट विल हैव 0.022 0.002 0.02 mm। सॉइल का डायमीटर, सॉइल का एक छोटा सा एक यूनिट जो होता है पत्थर मिट्टी का उसका यूनिट। सो फाइन सैंड, so you will have particle size in the range of 0.0 to 2.2 mm. For coarse sand 0.2 to 2 mm. There are different type of soils. Clay soil होता है, loamy soil होता है, slit loam soil होता है, sandy loam soil होता है, sandy soil होता है, calcareous soil. These are the types of soil. Okay. So a particular type of plant will grow in a particular type of soil. That's why soil parameter is important again. So clay soil मतलब क्या है, more than 20% clay होगा। loamy soil मतलब क्या, 30 to 50% clay। स्लिट सॉइल मतलब 20 to 30% clay। सैंडी लोम मतलब 10 to 20% clay। सैंड रहेगा इसमें, इसमें क्या रहेगा, सैंड। देन सैंडी सॉइल मोर देन 75%, 70% सैंड रहेगा। कैलकेरियस सॉइल मोर देन 20% क्या रहेगा, लाइम। सो इट इज नोन एज अ बेसिक सॉइल, ये बेसिक सॉइल, बेसिक सॉइल। सो टाइप ऑफ सॉइल, अगेन कल्टीवेशन of the plant depends on the different type, like variety of plant, plant का variety कौन सा है, ठीक है, method of sowing, method of growing, cultivation parameters, collection parameters. So all these factors will affect the ultimately the, the quality of that product which are going to obtain from that plant. Okay. Now general methods for pest control, cultural method is there, physical method is there, biological method is there, and chemical method is there। कल्चरल मेथड मतलब क्या है कि आपने एक क्रॉप ले लिया, एक क्रॉप ले लिया, उसके बाद में ना चेंजिंग द टाइम ऑफ सोइंग एंड हार्वेस्टिंग मेंटेनेंस, चेंजिंग द टाइम ऑफ सोइंग एंड हार्वेस्टिंग, मेंटेनेंस ऑफ स्टोरेज, स्पेशल कल्टीवेशन मेथड्स, प्रॉपर क्लीनिंग एंड यूज ऑफ ट्रैप क्रॉप्स फॉर एंड रेजिस्टेंस वैरायटीज। ट्रैप क्रॉप्स मतलब क्या है, फॉर एग्जांपल आपने कॉटन लगाया, तो कॉटन के जो फील्ड के लास्ट वाला जो पार्ट होता है एंड, फील्ड्स के जो लास्ट वाले दो रो हो गए, इससे पूरा अपना ये फील्ड है, मैं क्या किया यहाँ पे सब कॉटन लगाया, बट ये जो यहाँ का जो रो है कॉटन का, वहाँ पे क्या आ गया आपने ट्रैप्स, ऐसे प्लांट जो अगर पेस्ट आ गए ना तो इसके ऊपर आ जाएगा और ये पेस्ट इसको आके मर जाएगा, तो आपका बाकी का जो प्लॉट है वो बच जाएगा। सो यूजिंग अ trap crops. In resistant variety, so use the resistant variety, pest resistant variety. So known as a cultural pest control. Physical pest control means mechanical control of pest, utilization of physical factors like temperature, less oxygen concentration, humidity, passing the carbon dioxide. मतलब आपके प्लॉट में क्या करना है आपको कि टेम्परेचर ऑप्टिमम रखना है, ऑक्सीजन लेस रखना है. अगर ऑक्सीजन कम रहेगा तो ऑब्वियसली वो पेस्ट वहाँ पे मतलब ऑब्वियसली वो कीड़े वगैरह वहाँ पे ग्रो नहीं होंगे. बायोलॉजिकल पेस्ट कंट्रोल यूजिंग द प्रेडिटर्स मतलब क्या है, जैसे पर्टिकुलर टाइप के कीड़े होते हैं, वो कीड़े क्या करते हैं कि जो पेस्ट वगैरह आता है उसको खा लेते हैं. तो यूजिंग द प्रेडिटर्स, parasites, pathogens, sterilization, genetic manipulation, and pheromones. Okay? And chemical method for pest control. Use of pesticides, herbicides, and anti-predator. ये तो mostly अपन करते हैं. Now, next we will see metabolic pathways in the plant. Metabolic pathways in the metabolic pathways. Again, it is important. So, metabolic pathways for how there is a metabolism in the plant, how what are the primary metabolites, what are the secondary metabolites, how they are generated. Okay, so on that part also they have already asked one or two time questions. That's why I have included this part. So before this there was no this. Today I have included. So metabolic pathways in the higher plant and their determination. ठीक है, क्योंकि ये सॉफ्टवेयर का थोड़ा सा इश्यू चल रहा है, वो मिल नहीं रहा है, अभी ये पुराने लैपटॉप पे ही चल रहा है. Now, all living beings, okay, all living, whether it is a animal or whether it is a plant, सभी जो जिंदा है ना, so all living being needs a various number of organic molecules for the survival. So all living beings needs a various organic molecule for survival purpose, like for respiration, for formation of building blocks, for the production, etc., etc. So every living being, every living organism requires a organic molecule for survival purpose, like respiration के लिए आपको लगेगा, for formation of building blocks तो अमीनो एसिड लगेंगे, in case of plant कार्बोहाइड्रेट लगेगा, प्रोटेक्शन के लिए कुछ केमिकल्स लगेंगे, जैसे in case of plants अल्कोलाइड्स होते हैं, अल्कोलाइड्स मोस्टली आर यूज्ड फॉर द प्रोडक्शन ऑफ प्लांट. ओके, कार्बोहाइड्रेट, लिपिड्स, प्रोटीन्स एंड न्यूक्लिक एसिड, फॉर एग्जांपल दिस. For the synthesis of these molecules, for the synthesis of carbohydrate, lipids, proteins, and nucleic acids, and for other components, a series of chain reaction occurs in the organism. For the survival purpose, you need a certain biomolecule, certain organic components, and they are synthesized within your body. And for their synthesis, there is a series of chain reaction that occurs in the every individual. Okay? And the long chain of these reaction, the chain of these reaction or the path of these reaction, they are known as a metabolic pathways. जो रिएक्शन का चेन है या फिर आपका जो पाथ है रिएक्शन, उसको क्या बोलेंगे? ओके, उसको बोलेंगे अपन मेटाबॉलिक पाथवेज. Are you able to see the screen? Screen is there. व्यवस्थित. Series of chain reaction that occurs inside the organism for the production of these biomolecules, carbohydrate, protein, lipids, nucleic acid, or the organic components. Okay, they are known as the metabolic pathways. Now, though the organisms they are very much different from individual. है ना, आप अलग हो, मैं अलग है, गोल्ड का ठीक है, आपका रोज का प्लांट का मेटाबॉलिज्म अलग है, आपका पपाया का प्लांट का मेटाबॉलिज्म अलग है. Though organisms are very much different in their characteristics, but general pathway for production of carbohydrate, lipids, proteins, nucleic acid, it is very similar to each other. Now, these common pathway, which are common pathway in the plant as well as okay, all varieties of plant and animals, they are known as a they are known as a they are known as a primary metabolism. It is known as a primary metabolism. So, though each and every living being is different, for the pathway or for the reaction chain of reaction for the production of carbohydrate, protein, lipids, nucleic acid. They are very much similar in the individual. This common pathway in the these common pathway in the all organisms, they are collectively known as a primary metabolism. ये जो pathway होते हैं इनको बोलते हैं primary metabolism, and the product which are formed in the primary metabolism, they are known as a primary metabolites. इसको बोलेंगे primary metabolites. ठीक है, कार्बोहाइड्रेट, प्रोटीन, न्यूक्लिक एसिड, इसके लिए जो पाथवेज होते हैं सब में कॉमन होते हैं, and they are known as a primary metabolic pathway, and the product obtained after that primary metabolic pathway, it is known as a primary metabolites. प्राइमरी मेटाबोलाइट, तो इसमें क्या आएगा, प्राइमरी मेटाबोलाइट में क्या आएगा, यस एनीबडी, प्राइमरी मेटाबोलाइट में क्या क्या आएगा मोस्टली, okay, so all biomolecules they comes under the primary metabolite. Okay, apart from primary metabolites, okay, apart from primary metabolic pathway, there are some more compounds which are less distributed in the nature. ये जो है आपके, they are most abundant in the nature, they are widely distributed. But there are certain other pathways, other compounds which are less distributed in the nature, and these compounds are known as secondary metabolite. These compounds are known as a secondary metabolite in the plant. The pathway for production of secondary metabolite is known as secondary pathway. What are the secondary metabolites? So all, for example, your carbohydrate, all, for example, all organic compound like alkaloids, glycosides, terpenoids, tannins, volatile oils, right? Then your gums, all they are nothing but secondary metabolites. Some secondary metabolites they are produced in the plant for obvious reason. उसके एक reason होता है plant में, for the production like factor, like for the protection factor, protection factor like defense mechanism, pollination by insect. These are the secondary metabolite which show vast pharmacological activity. Secondary metabolite important है. Okay. Primary metabolite are not important from the plant. Secondary metabolite from the plant, they are important. They are having medicinal value. Okay. Secondary metabolite like your alkaloids, glycosides, terpenoids, tannins, volatile oils, they are not important because they have vast pharmacological. Now you can see in this what is meant by metabolism. So metabolism is nothing but a particular type kind of pathway. It may be either synthetic or it may be either catalytic. So anabolism is what? Anabolism, in the anabolism energy is utilized. Anabolism means what? Synthesis of building blocks from the monomers or simple units. For example, cellulose synthesis. Cellulose is a polymer of your carbohydrate. स्टार्च का ही पॉलीमर होता है सेलुलोज. उसको बनाने के लिए क्या है, उसको बनाने के लिए मेटाबॉलिज्म जो होता है उसमें एनर्जी खर्च होता है, एनर्जी यूटिलाइज्ड. So anabolic pathway. Catabolism means where there is energy production. Catabolism is energy production. Now in case of plants, plant में क्या होता है देखो, plant produces the energy. Plant will synthesize the carbohydrate. Plant क्या करते हैं, कार्बोहाइड्रेट बनाते हैं. कैसे बनाते हैं? By utilizing the carbon dioxide and water. So they uses the carbon dioxide and water and they produces the carbohydrate by using photosynthesis. Photosynthesis होगा यहाँ पे, बाद में पेंटोस फॉस्फेट पाथवे एंड बाद में बनेगा कार्बोहाइड्रेट. Plant produces the sugars. By photosynthesis. Now once there is a carbohydrate present in the plant, carbohydrate आ चुका है plant में, तो उसका मेटाबॉलिज्म हो सकता है. Carbohydrate is used for the metabolism. So ग्लाइकोलिसिस का एंड प्रोडक्ट होता है आपको पायरुविक एसिड. यू नो दैट. कार्बोहाइड्रेट से आपको बनेगा पायरुविक एसिड. दिस पायरुविक एसिड is converted to acetyl CoA. And acetyl CoA is known as central molecule and it is used for production of variety of compounds in the plant. So acetyl CoA से बनेगा आपका फैटी एसिड्स. फैटी एसिड कैसे बनता है? By acetate malonate pathway. Each and every single pathway we are going to see is just a primary information. From acetyl CoA also you are going to get polyketides, and from polyketides you are going to get aromatic compound. From acetyl CoA you are going to get terpenoids also, terpenoids through a acetate mevalonate pathway. This carbohydrate can be converted into aromatic compounds by using glycolysis, and also from the cyclic acid pathway aromatic compounds are generated. Now these aromatic compounds, there is a transamination or deamination, and you are going to get amino acids. Once you got amino acids, you know that from amino acids protein can be synthesized. From amino acids alkaloids can be synthesized in the plant. From amino acids only nucleic acid in the plant is synthesized. So it is a just overview of metabolism in the plant. This is metabolism of the plant. Next we'll see next will is cyclic acid pathway. Cyclic acid pathway. तो पहले आपको ये दिखा देता हूँ. Are you able to see the screen? दिखती screen दिखती का? So cyclic acid pathway. So क्वेश्चन आ चुका है इसके ऊपर. Amino acids are biosynthesized by following pathway. क्वेश्चन आ चुका है जीपीएट में. Amino acids कहाँ पे मिलेंगे? Amino acids are the intermediate which are formed in the cyclic acid pathway. You have to remember. Okay? Cyclic acid pathway. पहले अपन ये देख लेंगे एक ओवरव्यू, उसके बाद में आपको रिएक्शन बोलूँ. So erythrose 4-phosphate is a carbohydrate compound. Erythrose 4-phosphate and phosphoenol pyruvate. They will be condensed to form 3-deoxy D-arabino heptulosonic acid 7-phosphate. 3-deoxy D-arabino heptulosonic acid 7-phosphate. उसको बोलेंगे DAHP. So these two molecules, they are condensed to form a pseudoheptalic acid, and from that there is a conversion of this compound into quinic acid, dihydroquinic acid. From that plastic quinic acid. Now this 3-dihydroquinic acid is converted into 3-dihydrocyclimic acid. Cyclimic acid के बाद में 3-dihydrocyclimic acid से cyclimic acid बनेगा. बाद में cyclimic acid से 3-enoyl pyruvate cyclimic acid acetate 5-phosphate और 3-enoyl pyruvate cyclimic acid 5-phosphate का बनेगा बाद में corismic acid. Okay, और क्वेश्चन ऐसा ही था. Corismic acid is intermediate formed in the which of the following pathway. वहाँ पे pathways के नाम लिखे थे. cyclic acid pathway, acetate pathway, acetate mevalonate pathway. याद रखो corismic acid is formed in the cyclic acid pathway. From the corismic acid only you are going to get a anthranilic acid. From anthranilic acid you are going to get a amino acid known as a tryptophan. Okay, known as a okay. From the corismic acid only you are going to get a para-amino benzoic acid derivative, and after that this PABA will be converted to salicylate, and that's why in the plant also vinegar or extracts will have salicylate. Now from the pre-corismic acid only another pre-phenamic acid is formed, and from pre-phenamic acid you are going to get a aromatic amino acids known as phenylalanine and tyrosine. Once you get these amino acids, these are nothing but the biosynthetic precursor for. ये क्या होते हैं कि biosynthetic precursor होते हैं for the alkaloids precursor होते हैं. Cyclimic acid pathway for GPAT. Erythrose 4-phosphate and phosphoenol pyruvate, they will interact with each other and they are condensed to form a 2-keto-3-deoxy 7-phosphoglucoheptanoic acid, and it is catalyzed by enzyme known as a DAPH synthetase. Once you got a phosphoglucoheptanoic acid, so that is converted into 3-dihydroquinate. Phosphoheptoglutamate acid is converted into 3-dihydroquinate by the enzyme known as a dihydroquinate synthase. Okay, पहले किसमें the compound 2-keto-3-deoxy 7-phosphoglucoheptanoic acid, इसको बनेगा आपका 3-dihydroquinate from the by the enzyme dihydroquinate synthase. Last में nicotinamide adenine diphosphate NAD is used as a cofactor for this reaction. Next reaction क्या होता है? In presence of another enzyme known as the 3-dihydroquinate dehydratase. 3-dihydroquinate dehydratase. These 3-dihydroquinate is converted into dihydrocyclimic acid. 3-dihydrocyclimic acid. Then there is a phosphorylation with addition of phosphate group. ATP-based phosphorylation occurs of the product dihydrocyclimic acid, and इसका phosphorylation होता है and you will get a shikimate 3 phosphate in presence of enzyme shikimate kinase. बाद में phosphoenol pyruvate जो होता है, that phosphoenol pyruvate react with the shikimate 7 phosphate to form a 5-enol pyruvate shikimate 3 phosphate. 5-enol pyruvate shikimate 3 phosphate with the help of enzyme 5-enol pyruvate shikimate 3 phosphate synthesis EPSP. Next product is nothing but the corismate, which is formed from the 5-enol pyruvate shikimate pyruvate. So corismate is the product which is formed from the 5-enol pyruvate shikimate 3 phosphate by enzyme corismate synthesis. इसके बाद में pleasant rearrangement occurs with the corismate in presence of enzyme known as corismate mutase, and there is a formation of prefenamic acid. Prefenamic acid. From the corismic acid, various intermediate products like anthanolic acid, phosphoribosyl anthanolic acid were formed, which will last for the lastly will produce tryptophan. तो याद रखना है आपको, यहाँ पे 3 एसिड है, सिक्किमिक एसिड है, प्रीफेनेमिक एसिड है एंड कोरिज्मिक एसिड. एक बार ऐसा क्वेश्चन आया था कि सिक्किमिक एसिड, प्रीफेनेमिक एसिड, आई थिंक 2011 में आया था, व्हेन वी वाज फर्स्ट टाइम राइटिंग ए जीपैट एग्जाम. उस टाइम पे एक क्वेश्चन आया था कि कोरिज्मिक एसिड, सिक्किमिक एसिड एंड प्रीफेनेमिक एसिड, these are the 3 intermediate in the following metabolism. So सिक्किमिक एसिड पाथवे का ये मेटाबॉलिज्म में इंटरमीडिएट. तो ऐसा भी क्वेश्चन एक बार आया है, पहला कौन सा होता है. सो यू हैव टू रिमेंबर सिक्किमिक एसिड इज द फर्स्ट, देन कोरिज्मिक एसिड इज सेकंड, एंड देन प्रीफेनेमिक एसिड इज सिक्किमिक, कोरिज्मिक एंड प्रीफेनेमिक. ओके, यू हैव टू रिमेंबर. Prefenamic acid is converted to phenyl pyruvic acid or 4-hydroxyphenyl pyruvic acid, and which will lastly produce either phenylalanine and tyrosine. देखो यहाँ पे और एक बार. So ethers 4-phosphate and phosphoenol pyruvate will be combined together to form a 3-deoxy 3-arabino heptosulfonic acid 7-phosphate known as DAHP. Dehydroarabinoheptosulfonic phosphate. It's called the DAHP. Okay. Now from this, okay, it is catalyzed by DAHP synthesis enzyme. Okay. Then this DAHP is converted to 3-dehydroquinic acid by the dehydroquinate enzyme. Okay. Now from the dehydroquinic acid, this dehydroquinic acid you will get from that you will get a quinic acid only. ये इसका separately utilize होगा for production of secondary metabolite. From 3-dehydroquinic acid you are going to get 3-dehydroshikimic acid. 3-dehydroshikimic acid. 3-dehydroshikimic acid से बनेगा आपका shikimic acid. Shikimic acid से बनेगा आपका, there is a phosphorylation of this shikimic acid and 3-enoyl pyruvyl shikimic acid 5-phosphate. 3-enoyl pyruvyl shikimic acid 3-phosphate बनेगा. From that 3-enoyl pyruvyl shikimic acid 3-phosphate. 5-phosphate से बनेगा आपका कोरिज्मिक एसिड. कोरिज्मिक एसिड से डायरेक्टली एंथैलेनिक एसिड बनेगा, बाद में ट्रिप्टोफैन बनेगा. From the corismic acid, prefenamic acid is also formed. From prefenamic acid एक एंड पे आपका फेनाइलएलानिन बनेगा, एक एंड पे आपको क्या बनेगा, एक एंड पे आपको बनेगा टायरोसिन, और ये एंथैलेनिक एसिड जो है उससे बनेगा ट्रिप्टोफैन. दिस इज नोन एज सिक्किमिक एसिड पाथवे. वंस हो गए क्वेश्चन 2-3 टाइप के आ चुके हैं कि सिक्किमिक एसिड, कोरिज्मिक एसिड, प्रेफेनेमिक एसिड, these are the three acid intermediates which are formed in the following pathway. तो उसमें सिक्किमिक एसिड पाथवे आएगा आंसर. और एक क्वेश्चन आया था कौन सा कि aromatic amino acids they are the biosynthesized form which pathways in the plant. तो again सिक्किमिक एसिड पाथवे. Are you getting or not? समझ रहा है आपको? Yes, hello, are you getting? नहीं अगर समझ रहा तो बोलो, मुझे फिर से बोलो, बोलना है आपको फटाफट. If you are getting then okay, if you are not getting then just remind me. समझ रहा है आपको? Yes or no? What is the importance of this सिक्किमिक एसिड पाथवे? Importance क्या है इसका? Importance of सिक्किमिक एसिड पाथवे. So this pathway is very important to produce a phenylpropanoids. Phenylpropanoids, for example, phenylpropanoids means phenylalanine amino acid tyrosine. They are the main precursor for the synthesis of coumarin, lignins, tannins and flavonoids. तो जो सेकेंडरी मेटाबोलाइट्स होते हैं लिग्निन, coumarin, tannins, flavonoids वो कहाँ से आएंगे आपको phenylpropanoid unit से. Phenylpropanoid units मतलब क्या phenylalanine and tyrosine. They are synthesized in the cyclic acid. Then gallic acid is also biosynthesized from the cyclic acid pathway. There is a rearrangement which occurs of a 3,5-dihydroquinate shikimate dihydroshikimate in the presence of enzyme shikimate dehydrogenase. वहाँ से बनेगा gallic acid. Then aromatic metabolite and alkaloids are also formed by this cycle. That is the sequence of your shikimic acid pathway. Overall pathway क्या होता है shikimic acid pathway. Shikimic acid बनेगा, बाद में कोरिमिक acid बनेगा, बाद में प्रीपेनामिक acid बनेगा. प्रीपेनामिक acid से बनेगा phenylalanine and tyrosine. Tyrosine बनेगा, tyrosine से बनेगा अल्कोलॉइड. phenylalanine बनेगा, उससे बनेगा अल्कोलॉइड. प्रीपेनामिक एसिड से भी फिनाइल C3 कंपाउंड बनते हैं, उससे बाद में फ्लेवोनॉइड बनते हैं. सिनेमिक एसिड भी उससे ही बनते हैं, फिनाइल 3 कार्बन. यू कैन सी द समरी ऑफ सेम थिंग. शिकिमिक एसिड से बनेगा आपका क्लोरोजेनिक एसिड. क्लोरोजेनिक एसिड से उसका कोई ज़्यादा रोल नहीं है. इससे भी क्लोरोजेनिक एसिड से भी इट इज एक सेकेंडरी मेटाबोलाइट. बट शिकिमिक एसिड इज कन्वर्टेड टू कोरिमिक एसिड. कोरिमिक एसिड से बनेगा एंथ्रालिक एसिड, इससे बनेगा ट्रिप्टोफैन. ट्रिप्टोफैन से क्या बनता है देखो इंडोल अल्कोहॉल्स. इंडोल ग्लूकोज इनोलेट, इंडोल पाइडोलेक्सिल, ट्रिप्टामाइन, इंडोल एंड फिनोलिन अल्कोहॉल्स. कोरिमिक एसिड से बनेगा फिनाइललालानीन, उससे क्या बनता है फिनाइल इथाइलामाइन, सिनेमिक एसिड, देन आइसोक्विनिन एंड बेंजोइल आइसोक्विनिन टाइप ऑफ अल्कोहॉल्स. सिनेमिक एसिड से क्या होता है सिनेमिक एसिड से बनेगा फ्लेवोनॉइड्स एंड एंथोसाइनिन्स, लिग्निन प्रिकर्सर, coumarins, फाइटोअलेक्सिन, सैलिसिलिक एसिड. सैलिसिलिक एसिड से क्या बनता है कैटेकॉल बनता है, टैनिन्स बनते हैं, मिथाइल सैलिसिलेट, हाइड्रोक्सीबेंजिक एसिड. टायरोसिन बनेगा फेनिलएलानिन से. टायरोसिन से क्या बनता है अल्कोलाइड्स, साइनिन, ग्लाइकोसाइड्स, बीटा आयंस एंड मॉर्फिन. टायरोसिन से टायरामाइन बनता है. टायरामाइन से ओलेराइडिन नाम का अल्कोलाइड बनता है. पहले ठीक है. So Shikimic acid pathway leading to biosynthesis of diverse bioactive principles. Biosynthesis of flavonoids. Biosynthesis of flavonoids. Flavonoids क्या होते हैं? They are the phenylpropanoid unit containing. Okay. They are known as the flavonoids. They have the antioxidant activity in the body. Flavonoids are biosynthesized via condensation of shikimic acid and acyl polymalonate pathway. Phenylpropane, it is a cinnamic acid derivative. They are synthesized from shikimic acid, which act as a precursor in the polyketide synthesis. So flavonoids are also known as a polyketide. इसमें बहुत सारे कीटोन ग्रुप होते हैं. In this scheme, the 3 acetic acid or acetate molecules they are condensed. In the synthesis of flavonoids, यहाँ पे 3 acetate molecules they are condensed to form a phenylpropane. So 3 acetate residues are incorporated into a structure followed by ring closure. The plant biosynthesizes the different classes of flavonoids like flavonols, flavanones, flavonols, catechins, isoflavones, dihydroflavonols, anthocyanidins, and chalcones through subsequent hydroxylation and reduction. There are 3 acetate molecules which are condensed together. 3 acetic acid molecule condensed together to form phenylpropane, and then there is a ring closing, condensation as well as a ring closing. So you will get a polyketide. From that you will get a chalcone molecule. Now all flavonoids are the derivative of this chalcone, the flavonoid. Biosynthesis of glycoside.